less than 20 years. And this is a brief of the research in my group. We did some interdisciplinary research. Previous several works on this non-equilibrium physics combined with the stochastic process and also single-cell biology. Specifically, we did some research on stochastic mathematics and also stochastic thermodynamics and also single-cell kinetics. And very recently, we started to do some statistical inference. We do genetic disease diagnosis based on some genetic data, and also we do some single-cell data analysis based on some new category of single-cell sequencing data. And today, I will talk about one story about single-cell kinetics, also combined with non-equilibrium thermodynamics. And in the past more than 20 years, people know there are a lot of interactions among math, physics, chemistry, and biology. You can see the left one is a notice in the preceding of the notice of American Mathematical Society. It's about the mathematical biology. And also the middle one is 20 years ago, people had already realized that biology can be studied from a physical perspective. And also the right one is almost 10 years ago, I was invited to contribute a small essay in the Cell. They want to let several people from different backgrounds to talk about the research to biology. And my background is the stochastic process. So the first question I want to ask is which kind of physical or chemical process can be described by a stochastic process? To be briefly answer this question, it's all about the time scale and the spatial scale. Typically, the time scale is between millisecond and the microsecond, and the spatial scale is on the nanometer. It's just the time scale and the spatial scale of a single large molecule and also single cell. And also in experiments, we can observe the trajectory of a stochastic process. So the knowledge of a stochastic process can help us to analyze the data and discover some scientific laws. So today, this story is about HIV. HIV is a very dangerous virus to human being. And we all know it can infect the T cells. And also the T cell can, when it is affected by the HIV, it can become two states. One state is called a latent state. It's just be silent. And the other state is called active state. It can be activated and also to infect more T cells. So actually, now people have a lot of drugs that can kill the active state, but now they cannot just kill the latent state T cells be affected by the infected by the HIV virus. So that's a big problem for the HIV treatment. And also you can see there is some the figure B is about the genome of the HIV. The HIV genome can be inserted into a human genome. So the genome is a little bit complicated, but you can just realize that there is some promoter regime called LTR, and the LTR can be transcribed and translated into a protein called TAT protein. And this protein can have some positive feedback on itself. So it's just like autocatalysis, just the self-activated molecule. And also people want to measure the stochasticity of a single promoter. They found that the HIV promoter, the stochasticity within the HIV promoter is very significant. It's much higher than the typical human promoters. This stochasticity is measured by the CV square, that is the variance divided by the square of the mean. So what is the mechanism behind that? So almost less than 20 years ago, people already know that if you synchronized cell, T cell be affected by the HIV virus, you can see they can have very distinct two state. One state is keep into the inactive state, always transcribed and translated into the TAT protein. And the other is it can be suddenly go to the silent state. It is stopped there for a long time. So that is a big problem for this mechanism of the HIV, because people want to explain why there is some biomolecular distribution. If you look at the trajectory, you can see there is some the biomolecular distribution always there. So why there is two state of this HIV LTR promoter? People already know that. You can see this one. Let me see, there is no movie. So this one is people has measured in 2007. They want to see whether we can use a deterministic model to model it. And as you all know, if the positive feedback is very strong, for example, the H parameter is more than, for example, more than 2 or even 3, you can see that then you can easy to get this bistability. That means that if you solve the fixed points of this ordinary differential equation, you can see at least two stable points. But in experiments, they only measure the H as 1. That means the positive feedback is very weak. So people know there is no called deterministic bistability. That is, if you only use this ordinary differential equations, you can never see two spontaneously existed two fixed points. So there must be another mechanism. So people also observe that if you measure, because they know that the LTR promoter will have also two states. One is the active state, one is the inactive state. In the inactive state, it stops to transcribe and translation. So people already know. You can see this is 2012 paper. They saw that this LTR promoter will stay in the on state for almost less than 1 hour, then jump to the off state, and stay on the off state for several hours, then go back to on state. Because this gene state switching is very, very slow. So you can see it's almost like when it is activated, then there is a high peak, for example, around 100. When it is in off state, there's a peak at the 0 state. Because their transition is very slow, so the final distribution you see is actually the linear combination of these two different peaks. Then you can see just the coexistence of these two peaks. So that is something already know for many years. And what we really interested in this problem is because of the science paper published 10 years ago by Leo Weinberg's group in UCSF. They want to make some contribution to the HIV treatment. So as I have mentioned, the big problem to treat HIV is now the drug can only kill the active state cell, cannot kill the latent state cells. So if you have some small molecules, these small molecules are all being approved by FDA. So if you use some already known small molecule medicine, if you combine them with some HIV drug, can it trigger the latent state cells to be more active? If it can be triggered more active, then it can be killed by the drugs. But already we know the drugs, the existing antivirus drugs. So they scan almost all the small molecule medicine being approved by FDA. They find that there are two categories. One category is called traditional activator. It means that if you put this drug into the T cell, it will increase the transcription and the translation rate of this LTR promoter. That is the very traditional ones. Everybody know. But they already also find another category of small molecules. It's called noise enhancer. It cannot increase the mean expression level of this LTR. This is TATR protein, but it can increase the noise. So it has been summarized on the right-hand side. So if you put the drug in, you measure the GFP has been transcribed by this LTR, you can see the activator can increase the mean, but decrease a little bit of the noise, and the noise enhancer can keep the mean, but increase the noise. Previously, people only thought that the activator can play a role, but nobody know why the noise enhancer or whether the noise enhancer can have some role on this HIV treatment. So this is their main result. This figure, this red figure, you can see they combine one activator with a lot of noise enhancer. They see many or majority of the noise enhancer, when it combined with the activator, it can increase the something called reactivation ratio. That means it will increase, it will let more inactive state or more latent state cells just transit into the active state. That is a very general phenomenon. So we are very curious what is the mechanism behind that. So there is a summary of this phenomenon. You can see this activator can increase the reactivation ratio, but the noise enhancer itself cannot. But if you combine them together, it will have some significant drug synergy that can much better than you using activator itself. So what is the mechanism behind this phenomenon? Because it's a general phenomenon, so we thought maybe mathematical modeling can play a role. So we first use the well-established model, is also in Leo Weinberg group. It's a two-state model. The LTR promoter can have two states. One is the on state, one is the off state. When it is on state, it can transcribe and translate it into TAT protein, and there is some very weak positive feedback. And also, we based on science paper, we add two assumptions. One assumption is that the activator can increase the ratio, the parameter K on, but do not affect the K off. And the noise enhancer, it can simultaneously decreasing K on and K off, become much slower. So they can increase the variance, but keep the mean. All of these assumptions have some, at least a partial support from experiments. But the very key assumption is a third one. That is, when you put them together, the activator can kill the effect of a noise enhancer on K on. That means if you put them together, activator can increase K on and noise enhancer can decrease K off. But noise enhancer can never decrease K on again. We want to test whether this assumption in your science paper is correct or not. So we introduce a parameter called F inhibition. When it is 0, this activator do not inhibit noise enhancer. When it is positive, it can inhibit. When it is 1, this activator can completely inhibit noise enhancer. This model is is a stochastic model. We use chemical mass equation model. So this is the results. So there are two necessary conditions for this drug synergy. One is the parameter called K off. That is the rate for the LTR state transit from the off state to the on state. Previously, in their original model, it's set to be 0.01. It's an arbitrary state parameter. But we found out that this parameter is completely not correct. So you have to increase it at least to 0.8. Then you can see this drug synergy. And also this parameter is validated by the previously measurement in 2012, I have already shown. So in their model, they just have some qualitative model. So they do not really care about the real numbers. But in our model, we want to be more quantitatively. So this is the first necessary condition. And the other one is this inhibition parameter F, it should be 1 or not 0. When it is 1, you have this red region. This region, they can have some significant drug synergy. So based on the two-state model, we have already explained the drug synergy. But is that enough? No, because it seems to be very mystery, because the activator can inhibit noise enhancers functioning on K-on, no matter which noise enhancer. This noise enhancer is very diverse. They have different structure, they have different behavior, but why the noise enhancer can inhibit all of them? So we build a four-state model. We thought the two-state model is too simplified. So the four-state model means that when the LTR state, the off state have two different states, and the on state also have two different states. The LTR star is, you can think about this as the LTR bound with some transcription factor. The activator is always transcription factor. It can bond with the LTR. So the green one means the effect of the activator, and the purple one is the effect of the noise enhancer. So they actually act on the different pathways, not on the same pathway. So they do not have direct interactions at all. And this four-state model, we have also two different categories. One is under detail balance, is at the chemical equivalent, and the other is energy input. You have something drive this circle. Is in the undetailed balance. For example, it can be driven by the ATP hydrolysis or whatever. And you can put this four-state state into this two-state model, but the downstream part is almost the same. And you see what happens. First of all, if it is with detailed balance, there's no synergy at all. We did some simulation, and also you can just put an effective two-state model. We combine this two black one into a single one and two red one into a single one, and we use some mean first passage time argument to calculate effective K-on and K-off, and we calculate an effective inhibition parameter. You can see the parameter is always 0. So there is no synergy. It's not a strange one, because the two-state model says that if this parameter is 0, then there is no drug synergy. And this is not only because of the simulation, because it's generally valid. It is a consequence of detail balance condition. We can actually prove that. It's very easy. But what if we break the detail balance? So we first of all, we added energy into a single transition rate. That is, there's 8 of them. You can put the energy into each one. And you can see, no matter you put it on which one, you can have this drug synergy as long as there is a probability flux clockwise. If you drive this probability flux counterclockwise, you can never have this drug synergy. And this can be proved. We just build a mathematical theorem, says that if the clockwise flux is positive, if and only if you calculate the stationary distribution of this on state, it with the A activator together with noise enhancer is larger than only with activator. It means drug synergy. So drug synergy, if and only if the probability flux is clockwise. But we can only produce somehow very weak drug synergy. Sometimes you cannot use your eyes to clearly see. So we want to see why there is some very significant drug synergy. How to produce very strong drug synergy? It's very simple, because when we look back into our several successful examples, when we break one transition rate, then there is a little bit strong drug synergy, and we clearly see why this why the mechanism, clearly see the mechanism behind this phenomenon, because the probability flux is clockwise. So if the LTR want to transit from the off state to the on state, it will always go from the right hand side. And when it want to back from the on state to the off state, it will transit from the left side. So this purple one, the thin, very thin purple one, actually, because the probability flux is clockwise, so this purple one is actually the effect of the noisy enhancer on the tail on. But you never reach this path, so it'll be inhibited. So enlightened by this, we just separate our energy into two different transition, but there are some tip invariant. We put beta 1 here and beta 2 here. And you can see when we tune beta 1, you can see there's some optimized synergy here. And this is the jet synergy on the stationary distribution, but in the experiments, they do not measure the stationary distribution. They just put the cell into a tube and see, for example, when you wait several hours and you see the ratio of this latent state to the active state. So we also simulated that, mimic the experiments. You can also see there's some jet synergy here. And we pick a random pick. We do not pick the optimized one. We just pick beta 1 equal to beta 2 equal to 5. And to reproduce all the results before, we can see the jet synergy very significant. You can see if you use the P on the stationary distribution as a measure, you can see it just previously, if you only have activator, it's also maybe 12%, but now it have nearly 25% or more than 25%. If you see the reactivation ratio, previously it's about 7%, and now it's about the doubles, always double, 14 or 13%. So, and also you can also use the effective two-state model, use the mean first passage time argument. You can calculate the effective T on and T off, and you can also calculate the effective inhibition parameter F. You can see it's always 1. It's consistent with the previously two-state model. And finally, in order to validate our model, we also use our model to produce all the previously known results about this HIV virus. And you can see we can produce almost all the important properties of this promoter. So that means that our model not only can explain this specific drug synergy, we can explain all the previous results about this HIV virus. So here is a summary. We uncovered 2 necessary conditions for this noise enhancer and activator drug synergy. And in order to explain why there is some very strange assumption on this two-state model, we propose a 4-state model with both transcription activation and positive feedback. And we show that detailed balance model can never produce drug synergy. And if you properly break in detailed balance with the sufficient amount and the proper direction, you are able to generate very strong drug synergy. So we explain the result proposed 10 years ago in experiments. So here's the acknowledgement. I want to send my collaborator, Professor Lei Zhang, also my colleague, Lei Zhang, and also the Dr. Xiaolu Guo, and also 2 students. Both of them are now in United States. When they did this project, they are undergraduate students. So it has been published 2 years ago. So thanks a lot.